Hello friends. Now I am going to discuss about first topic of unit first, herbs as raw materials. See, this is the syllabus of your this first topic. Definition of herb, herbal medicine, herbal medicinal product, herbal drug preparation, source of herb selection, identification and authentication of herbal materials, processing of herbal raw material. What is herb or define herb? Herbs generally refers to the leafy green or flowering parts of a plant, either fresh or dried, while spices are usually dried and produced from other parts of the plant, including seeds, bark, roots, and fruits. In general use, herbs are plants with savory or aromatic properties that are used for flavoring and garnishing food, for medicinal purposes, or for fragrances, excluding vegetables and other plants consumed for macronutrients. Culinary use typically distinguishes herbs from spices. In short, herbs are used in the form of different parts as seeds, barks, roots, and fruits. They may be fresh as well as dried and spices. Usually they are dried. So collectively, so these called as herbs. What is herbal medicine? Herbal medicine is the use of plants to treat diseases and enhance general health and well-being. Herbs can interact with other pharmaceutical medications and should be taken with care. Herbal medicine is also called herbalism, in which the study of pharmacognosy and the use of medicinal plants. Plants have been the basis for medical treatment through most of human history, and such traditional medicine is still widely practiced today. Modern medicine means allopathy medicine makes use of many plant derived compounds as the basis for evidence based pharmaceutical drugs. Although herbalism may apply modern standards of effectiveness testing to herbs and medicines derived from natural sources, few high quality clinical trials and standards for purity or dosage exist. The scope of herbal medicine is sometimes extended to include fungal and bee products as well as minerals, shells, and certain animal parts. In short, modern medicine or allopathy medicines they are derived using plant based products or compounds. This is according to WHO, more than 80% people in the world they rely on plants or their products. So, as per WHO, more than 30% prescriptions they derived through plant based entity or products. It includes fungal and bee products as well as mineral shells and certain animal parts. Herbal medicine is also called phytomedicine or phytotherapy. Paraherbalism is also one of the concept related to herbal medicine. Paraherbalism is the pseudo scientific use of extracts of plants or animal origin as supposed medicine or health promoting agent. Paraherbalism differs from plant derived medicines in standard pharmacology because it does not isolate or standardize biologically active compounds, but rather relies on the belief that preserving various substances from a given source with less processing is safer or more effective, for which there is no evidence. There is no any clinical trial like modern medicine or synthetic agent. Just they preserve or use as per the ethnomedicinal claim or the information is available in old books and treatises. There is no any clinical evidence. Okay, only as per the information available in old books, treatises, or according to ethnomedicinal claim. Okay, some medicines, plant derived medicines, are used to treat diseases or disorder or health promoting agent. It's called paraherbalism. Herbal dietary supplements most often fall under the phytotherapy category. Nutraceuticals or herbal dietary supplements is used again to prevent, mitigate, or treat diseases or disorder. Herbal dietary supplements or nutraceuticals they also come under category of phytotherapy or phytomedicine. What is herbal medicinal product? Herbal medicinal products are defined as any medicinal product exclusively containing as active ingredients one or more herbal substances, one or more herbal preparations, or a combination of the two. Given the particulars of herbal medicinal products, a committee for herbal medicinal products was established at the European Medicine Agency. The HPMC means Herbal Medicinal Product Committee. The HMPC has been tasked with establishing monographs for traditional herbal medicinal products and proposing a list of herbal substances or medicine which have been in medicinal use for more than 30 years and are therefore not considered to be harmful under normal conditions of use. In short, to prepare a list of medicinal products or herbal medicine, those are using more than 30 years to treat, to prevent or mitigate diseases or disorders and not considered to be harmful under normal conditions of use. So see here HMPS. So again the same thing, defined as any medicinal product exclusively containing as active ingredients one or more herbal substances or one or more herbal preparations or one or more such herbal substances in combination with one or more such herbal preparations. So this is additional slide only. Here the same information, but in short the information is mentioned here. Okay, so see here according to law on pharmaceutical products for human use, so directive 2001 oblique 83 oblique EC means European Committee applies to HMP, and so see that requires a marketing authorization or registration before placement on the market in the European community. So see here, protect public health, harmonize authorization across the European Union or states, facilitate the authorization registration procedures of HMP. So this is about the herbal medicinal product. What is herbal drug preparations? The herbal preparations thus obtained includes extracts, decoctions, tinctures, essential oils and others. The processes involved include extraction, distillation, fractionation, concentration, fermentation, or other chemical or biological methods. So, in short, the preparations of herbs or plants are prepared using any physical process or any processes like extraction, distillation, fractionation, concentration, fermentation, or any other chemical or biological method or technique. It includes extracts, decoction, tinctures, infusion, essential oils, and others. So, see, this slide indicates different herbal preparations, herbal extracts. So, it is a liquid preparation of herbs that obtained from the plants, or extract out soluble medicinal components from the plant materials. Tincture, so it is prepared using alcohol as the medium. Elixir, hydroalcoholic preparation. Syrup, a concentrated tea with the addition of a sweeteners such as honey, maple syrup, or glycerin. Tea, herbal extracts using water as the medium. Infusion, a process of making tea using a steeping process. Brew, decoction. Liniment, liniment that prepared by using oils, turpentine oil or any other suitable oil, mainly used uh, to use as a counter irritant, means apply on an affected part of body just to relieve pain or inflammation. Ointment, a mixture of oil and tea that is usually that easily absorb into the skin, means apply on an affected part of body externally. Sol, compress. So there are different herbal preparations. Source of herb selections. There are mainly two sources. First, wild source, and second, cultivation source. First, wild source. Examples: forest, plains, river bank, etc. Means plants or herbs obtained from their natural habitat, niche, as such obtained from their region. Advantages of wild source: when we collect herbs as a medicinal products, or when we collect herbs for the study purpose. Advantages: economical, low labor cost, because there is no any supervision of man or no need of provide manures and all these things. So naturally simply collect from their wild region or the area, and then use for their further study or use as a medicine directly. Disadvantages of wild source: quality cannot be predicted. We cannot predict quality when we obtain herbs or the plants from their wild source. Growth and yield is not uniform. Not ensure their growth and yield uniformly, years to years. Modern scientific technique or methods cannot be applied to increase the yield and quality. So these are disadvantages or limitations when we collect plant material or the herbs from their wild source. Cultivated source. In this, systematically cultivated by modern scientific techniques under supervision of man or experts. Quality and purity is ensured. Regular supply of raw material is possible. Better yield can be get. Application of modern scientific techniques like tissue culture, genetic engineering, hybridization, germplasm. So these techniques we can apply to increase the yield and the quality of product. Crop planning means regular supply of raw material to the industry. It ensures the regular supply of raw material as a medicine or for preparing a herbal preparation to the industry. So this is about the source of herbs. Source of herb selection we can learn in different sense also. Means how we can select the herbs for the study. Herbs and their information is as used to treat diseases or disorder in human being mentioned in different books, old books, treatises, or even mentioned in the form of different remnants. So we can collect that information, or we can refer number of books. Even we can go through a database of plants. We can learn the nature of chemical constituents which are present in these plants, and on the basis of their chemotaxonomy, on the basis of their pharmacological action, on the basis of their chemical constituents, different parameters, different things we can consider while we select herbs for the study and for their use as a medicine to treat, to prevent or to mitigate diseases and disorder in human being. So see here criteria for the selection of reference substances and quality control of herbal medicine should take into account that various ingredients may have different levels of influence on the final quality, safety and efficacy. For this reason, the order of selection of the substances for identification and quantification should follow the rules presented below. First, if constituents with known therapeutic activity have been identified, they should be used as markers. Means we can find out the chemical nature or the chemical constituents in the herbs or the plants. These chemical constituents may be known or unknown. If they are known, so simply we can find out their therapeutic activity, and then we can go for their screening and their further study. Second thing, if the chemical constituents or their nature or their therapeutic activity is not known, so then we can go for their bioassay or for their pharmacological activity or the study. Means clinical, preclinical and the clinical study for finding the use or the nature of the chemical constituents as a medicine. If the above cases are not applicable, the identity and quantity of herbal materials, preparations and medicines may be established by the production process and by analyzing marker substances containing other characteristic constituents. If we don't know the use or the therapeutic activity of the chemical constituents, so we can establish the uh, pattern of chemical constituents. We can find out the entities or markers in the plant material, or we can do their structural elucidation and then go for their further study, means their pharmacological study and other study. So this is about the criteria for the selection of plant material or selection of herbs. Identification and authentication. In the last few decades, there has been an exponential growth in the field of herbal medicine. It is getting popularized in developing as well as in developed countries due to its natural origin and lesser side effects. In short, So there is increasing the use of herbs or their products as a medicine exponentially in the world. In olden times, Vaidyas used to treat patient on individual basis and prepare drug according to the requirement of the patient, but the scenario has changed now. Medicines are being manufactured on the large scale in pharmaceutical units, where manufacturers come across many problems such as availability of good quality raw materials, authentication of raw material, availability of standards, proper standardization methodology of single drugs and formulation, and quality control parameters. Identification includes four steps. First step, search for official and vernacular names of the drug using formulary, pharmacopoeia, monographs, or any official book. Second step, list the possible adulterants and substitutes. Find out which type of the adulterants or substitutes may be there. Again, that information, that adulterants or substitutes, find out using books, old books, pharmacopoeia. Third step, taxonomically identify crude drugs for reference. So find out the different parts of the plant, nature of these different parts of plant, fruits, leaves, flowers, their stem, shape of leaves. So these different parts, okay, taxonomy, species, genus, order, class, family. So simply find out, try to find out, and prepare their specimen for uh, deposit in the herbarium for future reference, for future correspondence. Prepare herbarium as per standard specification and the guidelines, and the herbarium specimen placed or deposited in the drug museum or the herbarium of Government of India. For example, Botanical Survey of India. Fourth step, pharmacognostical identification of official adulterants and substitutes. Then find out their botanical description, color, order, taste, size, shape, means their morphological features, characteristics, then physico-chemical parameters, chemical identification. So all these things are collectively called as identification of herbs or plants. Authentication. After identification, the specimen, sample or voucher specimen prepared, and then the same is to be deposited in the herbarium for their future correspondence. And authentication means on the basis of the taxonomic, macroscopic and microscopic studies, records should be maintained, means verify or authenticate the name of the plant or the scientific name of the plant. Okay, so they issue the certificate or give their name, so it's called authentication. And so that authentication process is done by experts or taxonomist or botanist or naturalist in the departments, in the research center. Record should be maintained for stage of collection, parts of the plants collected, regional status, botanical identities such as phytomorphology, microscopical, histological analysis, taxonomical identity, etc. So see here, plant material is to be collected, means sometimes random selection or sometimes systematically selection of plant material for their study on the basis of the chemical nature, or their ethnomedicinal claim. But here random approach means random selection of plant material or the herbs for their study. Sometimes taxonomic approach means find out the member of the same family, find out the member of means same type of the chemical constituent sometimes in the plant material. On the basis of that, then we can select any plant for their study. Then phytochemical, so already I told you. So chemical constituents means if the same type of the chemical constituents in the plant, then we can go for their further study. Then medicinal approach. Ethnomedicinal approach means what are the old claims in olden days or ancient time. What is the use of these plants or the parts of these plants for diseases or disorder. So on the basis of that, then we can go for their study. Then information manage approach. So again, the information that collected from different sources and database that prepared, and on the basis of that, then we can go for their study. So plant material means selection of plant materials on the basis of the different approaches. Then go for their extraction means after processing plant material, collection from authentic source, their authentication at Botanical Survey of India or at any uh, authorized center. Then go for their extraction. Different solvents of different polarities are used for the extraction of plant material. Then we can get different plant extracts. Then these plant extracts used for the isolation of phytoconstituents, isolation of pure compound. Then these pure constituents used for their pharmacological study means bioassay or their biological activity can screen out or reveal using animal models is called preclinical study. Okay, then uh, go for the toxicological study or go for their uh, lethal study means what is the lethal dose of that plant extract. So find out their toxic dose level or toxicity first in animal, then maybe in human being means for their clinical trials. So see here toxicology, then bioassay means their pharmacokinetic and pharmacodynamic study, okay, in animal. So that is called preclinical study, and then go for clinical study if it is success. Preclinical study is success in animals, so then we can go for their further study, and at the same time can find out or establish the structure of any known or unknown compound using different spectroscopical or chromatographic methods or techniques is called structural elucidation. And after structural elucidation, when one molecule or their basic nucleus so establish or when we learn, so then we can go for their synthesis, combinatorial chemistry, and many modern techniques or methods we can use for the synthesis of that compound. So this is about the systematic approach of plant material for their study. Pharmacognostical, phytochemical and pharmacological study of plant material. Thank you. Thank you one and all.